Welcome to Chapman Academy. This is a YouTube platform for you to grow and advance your research writing skill. I recommend you subscribe and share Chapman Academy with your friends and colleagues so that they can also join the movement and then advance their research writing skill. So I've been taking you through the various phases in clinical trial. So I took you through the phase O. Today I'm going to teach you how to use the phase 1 in your study. So with the phase 1 clinical trial, it is done after the laboratory and then animal study. So for normal circumstance, phase 1, phase 2, phase 3 is the normal process for clinical trial. For phase O to be done, it means that it is being conducted under emergency purposes. So I'm starting with the phase 1 study. So in the phase 1 study, it is usually involve humans. That is the first study to involve humans. After the laboratory and then the animal study, we proceed with the phase 1 study. And then they are done to give the highest dose. So before you can determine the maximum dose for a drug that someone can be taking, it is being detected at the phase 1 study. So it is done to detect the highest dose of a drug or vaccine that can be given to a patient. So here we give it bit by bit until we get to the highest dose that can be given without severe side effects. So we give it to see the highest dose that can be given without causing the people harm, giving them severe side effects that can compromise on their lives. And then the treatment has been tested and then proven positive at the lab and then in the animal studies. But some of the side effects are not known because we've not given it to humans to see what the treatment or the drug or the vaccine can pose on human life. So here we include humans, then we give them the dose, then we can identify the various side effects that can pose on the individual or the group of people who have been given the treatment. And this study helps to identify the best way of giving the new treatment because it provides the step-by-step procedures that can be followed to give the new treatment to the people or the patients. It is to get the right way or get the best decision of delivering the particular intervention. It is being detected at the phase 1 study because here you are now starting the clinical trial. So you make sure you put in all the necessary and the needed procedures that can be followed in order to give the treatment to the people, because here any little mistake that you do can cause severe harm to the people, because this is the first time of trying the vaccine or the intervention and then the treatment. So you need to make sure that all the needed procedures are being followed. And then also here, the first study, it recruits very few people. Recruit very few people to the study. And then the dose are given bit by bit. So if today we give the participants let's say 1 ml, then we see that, oh, okay, they've been able to withstand the 1 ml without no side effects. Then we give them the next day or the next time, we give them 1.5, then we add 1.5, 1.5, 1.5 until we get to the state that we can see that, okay, at this state they are developing some side effects, then we can know the limit that we can give to the drug. That's why every drug has its own dosage. Others are taken at 0.5 ml, 1.5 ml, 1 ml, 2 ml, 3 ml, 5 ml. So that is how drug is being calculated. It is given based on the side effects. So if they see that at this level, when you take, you cannot withstand it, then they will reduce it. So that is how in phase 1 of clinical trials, that's why we are being able to identify the maximum dosage that can be given. And then we look to see if we can get any side effects. So here we are only checking for minor side effects. We are just checking for minor side effects that can arise after giving the intervention to the people. And then the next participant, it will go higher, as I said. So this procedure continues until we find out that, okay, now this is the best limit for the drug, the best dose for the drug. So we should just keep it here, that everybody who is participant to this study will take 5 ml per day. So that is how they came about the dosage that can be taken for a particular drug every day or per month or depending on the purpose of the drug. So at this stage, they are also looking for what the drug does to the body and how the body also responds to the drug. You see, we treated this in phase O, because phase O, it is combining all the 3 processes together. This process too is in phase O. So we want to give the drug to the people, and then we know how the drug will work with the body, and then how the body too will respond to the drug or the intervention that has been given to the people. So at this stage, phase 1, the hallmark is safety. The main concern of phase 1 is safety. We are just providing the needed form of intervention that will provide the highest and then the optimum safety to the people. So at this stage, we are not looking for how the drug will treat the disease or the condition, or the intervention will work to improve the life of the people. But here, we just want to know how safe the intervention is to the people, how best we can give the intervention to the people. That is what we look out to. And then the research team, we look close to the people and watch if we can identify any severe side effects. So we look at them every day. We check them, their BP, we check their sugar level, we check their hemoglobin level to see if they are developing any side effect. So we put cautious attention on the participant. Checks, we run series of checks on them. And then here we recruit small number to this phase, until later when we are moving to the next stage, that is the phase 2, then we can add more people to this study. And then at this stage, it is very few people that might benefit from the intervention. Very few people that might benefit from the intervention. Introduce. We don't give placebo at phase 1, not at all. We don't give placebo. Everybody is taking the active treatment. Everybody is giving the active treatment. Then we follow them, because here we want to know the best way, the safest way of giving the intervention. So when you introduce placebo into the intervention, you might not get the actual result that you are looking out for. So placebo is not introduced at this stage of clinical trial. And then at this phase, it carries the highest potential risk upon all the medical and then clinical trials. Phase 1 carries the highest potential risk because now the drug has not been tested before. No human beings have used the drug or the vaccine before. So we don't know what the drug does and then how the body responds to it. It gives the highest potential risk. That's why we even recruit very few people that if there is an adverse condition, it can be managed, because when you recruit a lot of people and then there are adverse conditions, it becomes very difficult for you to manage. But at this stage, we recruit very few people, then we make sure that we put key attention on them, we follow them every hour, we check their BP, every hour we check their hemoglobin level, we check their sugar level, a series of tests on them, so that we can detect every hidden adverse condition on these people. So this is what we do at phase 1 study. At phase 1 study, our hallmark is to check for the efficacy and then the safety of giving the drug. The best way that you can introduce the intervention to the people without causing severe harm. So look out for the other phases, that is the phase 2 and then the phase 3 through to the phase 4. I've done a lecture on them that will help you to understand the phases in clinical trial. So thank you for being with Chapman Academy on YouTube. I recommend you subscribe and share Chapman Academy with your friends and family. Share with everybody to also learn and follow the movement, whilst we learn the best way we can develop our research writing skills. Chapman Academy, see you there.